So, I had this massive realization last night while going over today's material, and I just have to start here. Have you ever actually stopped to think about what it takes to keep a multicellular organism alive?
I mean, it's pretty staggering when you really break it down.
Right. Not just breathing and eating, but the sheer mechanical and chemical coordination required every single millisecond. And to really grasp the structural organization of animals and this whole core biological concept of the division of labor, you're going to have to start by picturing the alternative.
Which is the single cell, the ultimate solo act.
Exactly. Imagine a unicellular organism. Let's look at something like an amoeba. If you think about it, the amoeba is the ultimate one-man band. You know those street performers who have like a drum strapped to their back, a guitar in their hands, a harmonica around their...
Cymbals between their knees.
Yes. They're doing absolutely everything simultaneously. And that's the amoeba. It's digesting food, it's exchanging gases with its environment, it's moving, it's managing its internal water balance, and reproducing, all within the incredibly tight confines of a single microscopic cell wall.
It's an impressive feat of nature, honestly.
It is, but it hits a massive physical wall very, very quickly. You can only get so big when you're doing everything by yourself.
And that physical wall is the surface area to volume ratio, which is, frankly, the absolute governing law of microscopic life.
Right.
If an amoeba simply grows larger, its internal volume increases at a cubic rate, but its outer surface area, the actual membrane it uses to absorb oxygen and expel waste, that only increases at a squared rate.
So the inside gets too big for the outside to handle.
Exactly. Eventually, it gets too thick. The oxygen cannot diffuse quickly enough to reach the center of the cell, and the metabolic waste can't diffuse out fast enough. The amoeba would literally suffocate on its own waste.
Which is terrifying.
It is the fundamental problem of life. How do you scale up? How do you build a larger, more dominant organism when the physics of a single cell strictly limit its size?
And the answer, of course, is multicellularity. But, this is the key, you can't just glue a billion amoebas together and call it a day.
No, because the cells on the inside of that cluster would still starve and suffocate. They wouldn't have access to the outside environment.
Right. Which brings us to the core essence of this topic. To build a complex animal, you need specialized transport systems. You need structural scaffolding. You need dedicated engines for movement and a highly complex wiring network to coordinate the whole thing.
Division of labor.
Division of labor. By splitting up the work, cells can specialize. They drop the drums and the harmonica and they just focus entirely on mastering the violin. They become incredibly efficient at one specific task rather than being mediocre at a dozen.
That's a great way to put it.
And for you listening today, that is exactly what we are going to unpack in this deep dive. This isn't just going to be a dry list of anatomical parts. We are going to build a clear, logical mental map of how simple cells organize into tissues and how those tissues are engineered to solve the real-world physical problems of survival.
And we're going to track that engineering through a very specific evolutionary progression. We'll establish the 4 fundamental building blocks of all animal tissues first. Then we will apply that knowledge to 3 major evolutionary stepping stones.
The earthworm, the cockroach, and the frog.
Exactly. And for those of you preparing for competitive exams like Neet, listen closely. This progression is crucial. Examiners don't just test your ability to memorize fancy Latin names. They test your understanding of why a specific structure exists in a specific animal and how it differs from human anatomy.
Oh, absolutely.
So we are going to map out the exact traps, the high-yield concepts, and the common mistakes that catch students off guard year after year.
Let's start with those building blocks then. When you look at the sheer complexity of a vertebrate, a frog or a human, it seems like there must be thousands of different base materials involved.
It does look that way from the outside.
But the reality is surprisingly elegant. There are only 4 fundamental pillars of tissue that make up everything in a complex animal: epithelial, connective, muscular, and neural tissues.
Just 4.
Just 4. If you think of building a skyscraper, you really only have a few base materials.
Yeah.
You have your drywall and paint to create rooms and boundaries. You have your steel scaffolding for support. You have the engines running the elevators, and you have the electrical wiring coordinating the whole building.
That is your body right there.
Let's look closely at that first pillar, the drywall and paint, epithelial tissue.
Epithelial tissue is best understood as the body's ultimate border patrol and inner lining. The defining physical characteristic of epithelium, and you need to visualize this, is that the cells are compactly packed together with almost 0 intercellular matrix between them.
They are standing shoulder to shoulder.
Exactly. guarding the borders of the body and lining the internal organs. Functionally, we divide them into two broad categories, simple and compound.
So simple is just one layer.
Simple epithelium consists of just one single layer of cells. Yes, you use a single layer when you need things to pass through easily, like oxygen diffusing into the blood or nutrients being absorbed in the gut.
Make us.
Compound epithelium, however, has two or more layers of cells. Its primary role is not absorption at all. It's about protection against chemical and mechanical stress. Our skin is a perfect example of a compound epithelium.
It makes perfect sense. Form completely follows function here. Now, if we zoom in on the simple epithelium, it divides further into three distinct shapes, right? Squamous, cuboidal, and columnar.
Correct. And again, the shape dictates the job.
Squamous epithelium is made of a single, incredibly thin layer of flattened cells with irregular boundaries. Think of paving stones, or actually think of a fried egg where the yolk is the nucleus.
That's a brilliant visual.
Because they are so incredibly thin, they are physically perfect for forming a diffusion boundary. If you need a gas or a liquid to cross a barrier rapidly, use squamous cells.
So you find them lining the microscopic air sacs of the lungs, the alveoli where oxygen needs to enter the blood as quickly as possible, and you find them forming the walls of the blood vessels themselves.
Then we step up in size to the cuboidal epithelium. As the name suggests, these are cube-like cells. If squamous cells are flat paving stones, cuboidal cells are like little ice cubes.
They're thicker, which means they have more internal cellular machinery.
Right, you typically find these in the ducts of glands and the tubular parts of nephrons in our kidneys. Because they have more internal volume, their main gig is active secretion and absorption, which requires cellular energy rather than just passive diffusion.
And finally, we have the columnar epithelium. These are a single layer of tall, slender cells, like columns or skyscrapers, with their nuclei usually pushed down right at the base.
You find these lining the stomach and the intestines, where they're heavily involved in secreting digestive enzymes and absorbing massive amounts of nutrients.
But this is where the basic definitions end and the high-level exam mechanics begin.
Oh yeah, here we go.
Examiners don't just ask you to identify a cube-shaped cell. That's too easy. They want to know how these cells modify their outer surfaces to perform highly specialized jobs. And this leads us right into the first major trap.
I have this started in my notes.
This is a classic high-yield Neet focus. Examiners love to target the microscopic modifications of the apical surface, the top surface of these epithelial cells. So here's trap number one. The proximal convoluted tubule, or PCT, of the nephron in the kidney.
Okay.
The exam will ask you for the specific tissue lining the PCT. The correct answer is not just cuboidal epithelium. It is brush-bordered cuboidal epithelium. The apical surface of these cells is covered in microvilli.
Let's visualize those microvilli for everyone listening. They are tiny, microscopic, finger-like projections of the cell membrane extending outward. And why do they exist? It goes right back to what we've discussed about the amoeba and the surface area to volume ratio.
The physics problem again.
Exactly. The kidney's job, the PCT, is to reabsorb massive amounts of water and nutrients back into the blood so you don't pee them out. If the top of the cell was just flat, there wouldn't be enough physical space for all the transport proteins needed to pull those nutrients back in.
Right.
By folding the membrane into thousands of tiny fingers, the microvilli drastically increase the effective surface area for absorption without increasing the overall size of the cell.
It's beautiful engineering. Microvilli are entirely about maximizing absorption area, but, and here is trap number two, students constantly confuse microvilli with cilia.
Yes, ciliated epithelium.
If you have tall columnar or cuboidal cells that have cilia on their free surface, their anatomical purpose is entirely different. Cilia are not folds of the membrane for absorption. They are active, hair-like structures powered by the cell's cytoskeleton. They move. They move. They act like a synchronized rowing team, constantly beating in a specific direction. Their job is to physically move particles, or sweep mucus, over the surface of the epithelium.
So if microvilli are stationary antennas absorbing signals, cilia are brooms sweeping the floor.
Exactly.
Where do we find these brooms? The anatomy shows they're mainly present in the inner surface of hollow organs where things need to be pushed along. They are in our bronchioles, sweeping mucus and trap dust upward and out of our respiratory tract so our lungs stay clear.
And crucially, they are in the fallopian tubes.
Ah.
This is the detail examiners absolutely love to test. An unfertilized egg cannot swim. It has no flagellum. It relies entirely on the ciliated epithelium of the fallopian tube to create a current that sweeps the ovum down toward the uterus. If you mix up the microvilli in the kidney with the cilia in the fallopian tubes on exam day, you will lose marks. It is a fundamental distinction to lock into your memory.
Microvilli absorb, cilia sweep. Got it. Now, while we're talking about these epithelial cells standing shoulder to shoulder, we have to look at the structural engineering that keeps them together. You can't just have cells touching. They need to be physically and chemically anchored.
In nearly all animal tissues, but especially prominent in the epithelium, you find highly specialized cell junctions. They provide both the structural integrity and the functional links between individual cells.
I always fall back on the analogy of building a brick wall to understand these junctions. There are three main types you need to know. First, tight junctions. Imagine you're building a wall for a swimming pool.
You can't just stack the bricks.
Right, you don't just stack them. You need mortar between them to stop the water from leaking out. Tight junctions do exactly that. They fuse the plasma membranes of adjacent cells together so tightly that they stop substances from leaking across a tissue layer. It's the biological equivalent of the seal on a Ziploc bag.
A very vital protective mechanism. Your stomach is full of highly corrosive acid. It is the tight junctions between the epithelial cells that prevent that acid from leaking out into your abdominal cavity.
But tight junctions don't provide a massive amount of mechanical strength. For that, you need the second type, adhering junctions. If tight junctions are the waterproofing, adhering junctions are the heavy-duty cement.
They perform the actual cementing function to keep neighboring cells physically locked together.
They attach to the internal cytoskeleton of the cells, acting like biological Velcro or spot welds, ensuring that when the tissue stretches, like your skin or your heart muscle, the cells don't just rip apart from one another.
And finally, we have the most fascinating of the three, gap junctions.
Yeah, these are wild.
If you have your sealed bricks and they're cemented together, imagine drilling secret tunnels connecting the inside of one brick directly to the inside of the neighboring brick.
Just a straight tube through.
Yes. Gap junctions connect the cytoplasm of adjoining cells. They are made of specialized proteins that form actual channels between the cells, allowing for the rapid, unimpeded transfer of ions, small molecules, and sometimes even larger molecules.
The cells can communicate instantly without having to secrete a chemical into the extracellular space and wait for the neighboring cell to detect it.
Keep the concept of those gap junctions fresh in your mind, because they are the absolute secret to how the muscular system functions, which we'll touch on shortly.
Perfect.
But first, we need to transition to the second pillar of our structural map, connective tissue.
To scaffolding.
The scaffolding. This is by far the most abundant and widely distributed tissue in the body of any complex animal. Its special function is right in the name. It links and supports other tissues and organs.
Now, a defining characteristic of connective tissues, with one glaring, highly tested exception, is that the cells within the tissue secrete fibers of structural proteins.
Primarily collagen or elastin.
Right. These secreted fibers provide the tissue with strength, elasticity, and flexibility. Alongside the fibers, the cells also secrete a modified polysaccharide that acts as a matrix, or a ground substance. So, a connective tissue is essentially cells suspended in a web of their own structural fibers and jelly-like matrix.
But let's address that glaring exception immediately, because reframing this in your head is a major breakthrough.
Listen, this blew my mind. Blood is a connective tissue.
It is.
When we normally think of connective tissue, we think of structural scaffolding. We think of a tough tendon connecting a bicep to a bone, or the cartilage shaping our ears. We naturally visualize something solid, but blood is a fluid. It doesn't physically bolt the stomach to the spine, so why is it categorized here?
Because it connects the entire body, chemically and nutritionally.
Exactly. It's a fluid connective tissue containing plasma, which acts as the liquid matrix, along with red blood cells, white blood cells, and platelets. It is the circulating river that links the oxygen in the lungs to the muscles in your toes, carrying hormones, nutrients, and waste products. But crucially, blood cells do not secrete structural collagen fibers.
If they did, our blood would clot solidly in our veins. And that is exactly the distinction examiners want you to understand. Blood is a connective tissue of mesodermal origin, but it lacks the secreted structural fibers characteristic of the rest of the family.
The rest of the connective tissue family is incredibly diverse, and exams will ask you to differentiate between the solid forms. You have loose connective tissue, like the areolar tissue found just beneath our skin. It has cells and fibers loosely arranged in a semi-fluid ground substance, acting as a flexible support framework for the epithelium above it.
But the real high-yield focus is on dense connective tissue. You need to clearly map out the difference between dense regular and dense irregular connective tissue.
The mechanics of this are just brilliant. It's all about how the body handles physical stress. In dense, regular connective tissue, the collagen fibers are arranged in neat, parallel rows between many parallel bundles of fibers.
Because all the structural cables are running in the exact same direction, the tissue possesses incredible tensile strength, but only in that one specific direction.
This is exactly what you need for a tendon, which links a muscle to a bone, or a ligament, which links a bone to another bone. When you flex your bicep, the force pulls straight up. The parallel fibers of the tendon handle that vertical tension perfectly.
But if you were to pull a tendon sideways, it would tear relatively easily.
Contrast that with dense, irregular connective tissue. Here, the fibroblasts and the collagen fibers are oriented differently. They form a chaotic, overlapping, messy web of fibers running in all three dimensions.
Because the fibers point everywhere, the tissue isn't incredibly strong in just one direction, but it provides excellent generalized strength in multiple directions simultaneously.
This is the structural architecture of the skin. Your skin needs to withstand being pulled, twisted, and stretched from every conceivable angle without tearing. The dense, irregular web handles that omnidirectional stress perfectly.
It is pure mechanical engineering applied to biology. And rounding out the connective tissues, we have the specialized structural forms, cartilage and bone.
Cartilage has a solid, pliable, intercellular material that actively resists compression. The cells of cartilage, called chondrocytes, are enclosed in small cavities called lacunae within the matrix they secrete. It's tough but flexible, providing the cushioning between our vertebrae and the shape of our nose.
Bone, on the other hand, trades that flexibility for rigid strength. It has a hard, non-pliable ground substance, heavily enriched with calcium salts and collagen fibers. Its cells, the osteocytes, are also present in lacunae spaces. Bone provides the ultimate structural frame, supporting the body's weight and protecting the softer tissues like the brain and the lungs.
Which brings us to the active components of the body, the final two pillars, muscular and neural tissues, the engines and the wiring.
Muscular tissue is composed of long, cylindrical fibers arranged in parallel arrays. These fibers are essentially biological motors that contract and relax in a coordinated fashion to cause movement.
We categorize them into three types based on their structure and location. You have skeletal muscle, which is striated, meaning it has a striped appearance under a microscope due to the arrangement of its contractile proteins, and it is voluntarily attached to skeletal bones, like your biceps.
Then you have smooth muscle, which is unstriated, involuntary, and found lining the walls of internal hollow organs like the stomach, intestines, and blood vessels.
And finally, we have cardiac muscle, a highly specialized contractile tissue found only in the heart. And this is where we return to those gap junctions we discussed earlier.
Right.
If you think about the heart, it is a pump that has to contract millions of times over a lifespan, and it has to do so in a perfectly synchronized rhythm. If the cells at the top of the heart contract randomly while the cells in the bottom are relaxing, the pump fails and the organism dies instantly.
So how do millions of cardiac muscle cells agree to contract at the exact same millisecond?
The answer is in their specific tissue modifications. The plasma membranes of cardiac cells are fused together at cell junctions. But at some of these fusion points, they have specialized communication junctions called intercalated discs.
And an examiner will almost certainly test your knowledge of intercalated discs. These discs function precisely like the gap junctions you described in the brick wall analogy. They are the secret tunnels.
Because of these intercalated discs, the cardiac muscle cells are electrically coupled.
They function as a syncytium, a single unified functional unit. When the pacemaker of the heart fires, the electrical signal doesn't have to slowly navigate through the extracellular fluid to reach every cell. The ions flood instantly through those tunnels.
When one cell receives the signal to contract, its neighbors are instantaneously stimulated to contract as well. The entire chamber squeezes as one.
It's a chain reaction moving at lightning speed. It's beautiful.
Finally, we must map out the neural tissue. This is the intricate wiring network that exerts the greatest control over the body's responsiveness to changing conditions. The stars of the show, the main excitable cells that transmit electrical impulses, are the neurons.
But there is a massive trap here that completely catches students off guard.
This one got me too when I was mapping this out.
Yes, students almost universally fall for this trap. When we study the nervous system, 99% of our time is spent studying the anatomy of a neuron. We diagram the branching dendrites, we trace the long axon, we look at the synapses.
Right, so because of this intense focus, we naturally assume that neurons make up the vast majority of the physical mass of the brain and the spinal cord.
But that is anatomically incorrect. The research clearly shows that neuroglia, the supporting, protecting, and insulating cells of the neural system, make up more than 1/2 the total volume of neural tissue in our body.
The support staff literally takes up more physical space than the primary actors. The neuroglia are the cells providing the nutrients, clearing the waste, and insulating the axons to speed up the electrical signals. I'm definitely highlighting that detail. Over 50% of the volume is neuroglia.
Keep that anchor in mind.
Okay, so we have firmly established our mental map of the four tissues. Epithelial for boundaries and linings, connective for scaffolding, support, and fluid transport, muscular for the cellular engines, and neural for the instantaneous wiring.
Now, the natural progression is to look at how evolutionary pressures took these four basic materials and organized them into complex organs and organ systems in specific animals.
We're going to track this by looking at three distinct creatures, starting with a masterclass in elegant simplicity. Let's dig into the earthworm, scientifically known as Pheretima.
When analyzing the earthworm, you must start with its ecological niche. The earthworm is a subterranean invertebrate. It lives in the damp, dark soil. To understand its anatomy, the best mental map to hold in your mind is a continuous segmented tube within a tube.
Tube within a tube.
The outer tube is the muscular body wall, and the inner tube is the straight, uncoiled alimentary canal, the digestive tract. This entire organism is essentially a highly specialized biological machine designed for one single repetitive job: eating raw dirt, extracting the decaying organic nutrients from that dirt, chemically neutralizing the soil acids, and passing the processed earth out the back, thereby aerating and enriching the soil.
It is nature's plow. It really is a tube. Let's look at its morphology, that outer tube. The body is long, cylindrical, and visibly divided into over 100 short, remarkably similar segments called metameres. This segmented design is called metamerism, and it is crucial for their movement.
But there is a specific vocabulary trap regarding the very front end of this tube. You have the prostomium and the peristomium. They sound almost identical, but structurally they are very different.
The prostomium is not a true body segment, right?
Correct. It is a fleshy, muscular lobe that sits right at the front. It serves as a protective covering for the mouth, but more importantly, it acts like a physical wedge. As the worm moves through tightly packed soil, it uses this prostomium wedge to force open microscopic cracks in the dirt so the rest of the body can crawl through. It also contains sensory structures to help the worm navigate the dark.
And the peristomium.
The peristomium, however, is the actual first true body segment, the buccal segment, and it physically contains the mouth opening.
That distinction between the sensory wedge and the first true segment is a very common exam question. Moving down the body of a mature earthworm, the most prominent visual anchor you will find is the clitellum.
Oh, yes.
This is a thick, dark, prominently swollen band of glandular tissue that covers segments 14, 15, and 16. It is visually striking, and it functionally divides the worm's body into three distinct regions: the preclitellar, the collarelar, and the postclitellar segments. We will revisit exactly why this glandular band is so important when we discuss how they reproduce.
I always wondered how a worm actually moves. They don't have legs, and they don't have a hard skeleton to push against.
Right. They use a hydrostatic skeleton fluid pressure inside their body segments, along with specialized structures called setae. Setae are tiny S-shaped bristles embedded in epidermal pits right in the middle of each segment. The worm can extend these bristles to physically grip the walls of its dirt tunnel, or retract them to slide forward.
It's an alternating mechanism of gripping and stretching. But here is the specific detail to note. Setae are not found on every single segment. They are noticeably absent in the first segment, the last segment, and crucially, they are absent in the clitellum segments.
Excellent observation. Now let's cut into that outer tube and trace the inner machinery, starting with the digestive journey. Remember, the diet here is raw soil mixed with decaying leaves and organic matter.
The mouth in the peristomium opens into a short buccal cavity in segments 1 to 3. This leads into a muscular pharynx. From there, a narrow esophagus in segments 5 to 7 carries the soil down into a highly specialized organ, the muscular gizzard located in segments 8 and 9.
The earthworm doesn't have teeth to chew the leaves it eats. Instead, this thick muscular gizzard acts as a powerful internal grinding machine. It contracts forcefully, using the ingested soil particles themselves, like biological sandpaper, to pulverize the decaying leaves into a fine paste.
And from that grinding gizzard, the processed food enters the stomach, which spans segments 9 to 14. And this brings up a brilliant chemical adaptation.
Think about what the worm is eating. Decaying organic matter rotting leaves is highly acidic. It creates humic acid in the soil. If the earthworm just absorbed that raw acid, it would destroy its internal pH balance.
To survive, the earthworm must actively neutralize its food. It does this using specialized calciferous glands that are present in the stomach. These glands secrete calcium carbonate directly into the digestive tract, neutralizing the humic acid before the food moves further down.
This is a massive memory anchor for everyone listening. Stomach, calciferous glands, neutralizing humic acid.
That chemical neutralization is vital. From the stomach, the intestine begins at the 15th segment and runs straight to the end of the body. But here, we encounter one of the most elegant architectural solutions in invertebrate biology.
The typhlosul.
Yes. The earthworm is a long, narrow cylinder. It has severely limited internal space. How does it ensure it absorbs enough nutrients from nutrient-poor dirt when it can't coil its intestines back and forth like a mammal?
At the 26th segment, the anatomy radically changes. From segment 26 onwards, except for the very last 23 to 25 segments, the intestine features a prominent internal median fold hanging down from the dorsal wall. This massive inward fold is called the typhlosul.
The geometry of this is just incredible. By folding the intestinal wall inward, the typhlosul drastically increases the effective surface area for absorption within that confined cylindrical space.
It functions on the exact same physical principle as the microscopic microvilli we discussed in the kidney, just scaled up to a macroscopic organ level. It ensures that as the dirt passes through, the maximum amount of nutrients can be extracted.
It is a masterpiece of spatial engineering. Now, once those nutrients are absorbed across the typhlosul, they must be transported to the rest of the body. This requires a circulatory system.
The earthworm utilizes a closed circulatory system, meaning its blood is contained within distinct blood vessels, capillaries, and a heart, rather than washing freely through the body cavity. But there is a massive trap here regarding the respiratory pigment, the hemoglobin.
I have this circled in bright red. If you think about human blood, our hemoglobin, the iron-rich pigment that binds oxygen and makes our blood red, is packaged safely inside millions of individual red blood cells. But the earthworm does not package its hemoglobin.
Wait, really?
In the earthworm, the hemoglobin is dissolved directly into the fluid blood plasma. The blood glands, which are located in segments 4, 5, and 6, produce the blood cells and this free-floating hemoglobin.
So what do their blood cells do?
The blood cells themselves are actually phagocytic in nature. They don't carry oxygen. They act like an immune system, roaming around and eating cellular debris and pathogens. Do not fall for the trap of thinking an earthworm's blood cells function like human red blood cells.
The evolutionary logic there is fascinating. Dissolving hemoglobin directly into the plasma increases the viscosity of the blood, which requires a strong pumping mechanism. But it is an effective early evolutionary solution for oxygen transport in a closed system.
Moving on to how the worm handles internal waste, we look at excretion. The earthworm utilizes highly coiled microscopic tubules called nephridia. These structures regulate both the volume and the chemical composition of the body fluids.
Based on their anatomical location, they are categorized into 3 types, though they are fundamentally similar in structure. First, you have septal nephridia. These are attached to both sides of the intersegmental septa, the inner walls dividing the segments from segment 15 all the way to the end, and they discharge their waste directly into the intestine.
Second, you have integumentary nephridia, which are attached to the inner lining of the body wall from segment 3 to the last, and these open directly onto the outer body surface.
And finally, you have pharyngeal nephridia, which are found as 3 paired tufts clustered in segments 4, 5, and 6. The orderliness of this segmented creature is amazing. Every segment has its specific plumbing.
And that strict organization extends to its reproduction, which operates on a very counterintuitive mechanism. Earthworms are hermaphrodites. They are fully bisexual. Functional tests and functional ovaries are present in the exact same individual worm.
There are 2 pairs of tests neatly located in the 10th and 11th segments, and there's 1 pair of ovaries attached right at the intersegmental septum dividing the 12th and 13th segments.
The male pores, where the sperm exits the body, are located on the 18th segment ventrolaterally. The single female pore, where the eggs exit, is situated mid-ventrally on the 14th segment.
But here is the critical paradox.
Yeah.
Even though a single worm possesses everything required to reproduce, they do not self-fertilize.
And we have to ask why. Why have both sets of organs if you aren't going to use them on yourself? It comes down to the evolutionary necessity of genetic diversity. Self-fertilization is essentially genetic cloning. In a changing environment with evolving diseases, a population of identical clones is highly vulnerable to extinction.
So earthworms have evolved a behavioral mechanism to ensure cross-fertilization. They undergo a mutual exchange of sperm. During the mating process, two worms align themselves pointing in opposite directions. They juxtapose their opposite gonadal openings and physically exchange packets of sperm called spermatophores.
The worms store this received sperm in specialized internal sacs called spermathecae.
But wait, if they just swap sperm and crawl away, where do the baby worms actually develop? This is where that massive visual anchor we discussed earlier, the clitellum, comes into play.
Ah, the cocoon.
Yes. The thick gland cells of the clitellum secrete a viscous fluid that hardens into a protective casing a cocoon. The worm then shimmies backward. As the cocoon slides forward over the worm's body, the worm deposits its own mature eggs into it and then deposits the stored sperm it received from its partner into it as well.
Fertilization doesn't happen inside the worm. It happens externally, inside this chemically sealed cocoon, which is finally slipped off the head and deposited into the damp soil. Inside that protective incubator, the embryos develop. After about 3 weeks, the cocoon hatches, releasing tiny, fully formed baby worms directly into the dirt. There is no intermediate larval stage.
It is a brilliant reproductive strategy for a subterranean environment. The cocoon protects the embryos from drying out and from predators. Now, let's dramatically shift the environment. We are moving from the damp, protective, highly stable environment of subterranean soil up into the harsh, dry, open air.
We are stepping up the evolutionary ladder to examine an organism built to withstand almost anything. Let's dig into the cockroach, the ultimate survivor, scientifically known as Periplaneta americana.
If the earthworm is a soft, vulnerable tube, perfectly adapted to wet dirt, the mental map for the cockroach is a heavily armored, highly mobile nocturnal tank. It features an open-flow engine and an array of incredibly complex, segmented sensor systems.
They belong to the phylum Arthropoda, the largest phylum in the animal kingdom, and the class Insecta. They are nocturnal omnivores, meaning they scavenge for any organic matter under the cover of darkness. And their entire morphology is defined by their armor.
Exactly. To survive in a dry terrestrial environment, you cannot have a soft, moist body wall like an earthworm. You would desiccate and die within hours. The entire body of the cockroach is covered by a hard, brown exoskeleton made of a complex polysaccharide called chitin.
This exoskeleton prevents water loss and provides structural defense. But armor presents a physical problem. If you are encased in a solid shell, how do you move?
The evolutionary solution is segmentation and joints. In each segment of the cockroach's body, the exoskeleton is formed of distinct hardened plates called sclerites. The dorsal plates covering the back are called tergites, and the ventral plates covering the belly are called sternites.
And here's the mechanical genius. These solid plates don't fuse into one rigid box. They are joined to each other by a thin, highly flexible articular membrane known as the arthrodial membrane.
I like to think of a medieval knight's suit of armor or a modern spacesuit. You have the heavy steel plates protecting the chest and back, but at the joints, the elbows and the waist, you have flexible chainmail or rubber joints that allow the limbs to bend.
The arthrodial membrane is that flexible chainmail connecting the rigid tergites and sternites so the armored tank can actually scurry across your kitchen floor.
That is a perfect structural analogy. Let's examine the command center of this tank, the head. It is roughly triangular and hangs downwards, positioned at a right angle to the longitudinal axis of the body.
What's fascinating is its embryological origin. The head is not a single structure. It is formed by the fusion of 6 separate embryonic segments. Because it is connected to the thorax by a highly flexible short extension of the prothorax, the neck, the head possesses incredible mobility in all directions, allowing the cockroach to constantly scan its environment.
It features a pair of large compound eyes and a pair of thread-like antennae that arise from membranous sockets, packed with sensory receptors to monitor the environment for food and danger.
And when it finds that food, we look at the mouthparts, which are complex biological machinery pointing downwards. Unlike the simple mouth opening of the earthworm, the cockroach has appendages engineered specifically for biting and chewing solid food. It's like a biological Swiss Army knife.
There's an upper lip called the labrum and a lower lip called the labium, which hold the food in place. The heavy lifting is done by a pair of mandibles, hard, toothed structures that violently grind and crush the food. Then you have a pair of maxillae that act like auxiliary arms, manipulating the food and pushing it into the mandibles.
And right in the middle, enclosed within this cavity, is a median flexible lobe acting exactly like a tongue, the hypopharynx.
The mechanical coordination of those mouthparts is remarkable. Moving posterior to the head, we find the thorax, which is the muscular engine room for locomotion. It is strictly divided into three segments: the prothorax, the mesothorax, and the metathorax. Each of these three segments bears a pair of jointed walking legs.
But the thorax also houses the flight apparatus. The wings are a defining evolutionary feature, but they are specialized. The first pair of wings, the forewings, arise from the mesothorax. These are called tegmina.
They are dark, opaque, and leathery. They are not used for flying. Their primary anatomical job is to act as a protective sheath, covering and shielding the delicate hind wings when the insect is at rest.
The actual mechanics of flight are powered entirely by the second pair of wings, the hind wings, which arise from the metathorax. These are transparent, membranous, and highly aerodynamic.
Following the thorax is the abdomen, which consists of exactly 10 segments in both adult males and females. But this brings us to one of the most frequently tested concepts in insect anatomy. We have arrived at the high-yield, neat focus regarding sexual dimorphism.
Examiners love to test if you can distinguish a male from a female cockroach just by examining the external anatomy of the final abdominal segments. There is a strict, unbreakable rule you must memorize.
Yes, and the distinction revolves around two specific structures. Here is the rule. Males possess a pair of short, thread-like structures projecting from the ninth sternite called anal styles. Females do not have anal styles, period.
However, it is vital to know that both sexes possess a pair of jointed, filamentous sensory structures called anal cerci attached to the 10th segment. The anal cerci act as sensitive wind detectors, alerting the cockroach to approaching predators from behind.
So anal styles equal males only. Anal cerci equal both sexes. If a multiple-choice question asks which structural feature is unique to the male cockroach, the answer is the anal styles. Do not let the examiner confuse you by offering cerci as an option.
Anal styles equal males. Got it.
Now, let's crack open the armored tank and explore what you call the open engine, the internal anatomy. We start with the digestive system, the alimentary canal, which is divided into three distinct functional regions: the foregut, the midgut, and the hindgut.
The entire foregut is lined internally by the protective cuticle. Food is chewed by the mandibles, enters the mouth, passes through a short tubular pharynx, and travels down a narrow esophagus. It then dumps into a large, thin-walled sac called the crop, which is primarily used for storing the food. But the real mechanical processing happens next.
From the crop, the food enters the proventriculus, or the gizzard. We saw a muscular gizzard in the earthworm, but the cockroach's gizzard is heavily armed. It consists of an outer layer of thick, powerful circular muscles and an internal cuticle that is modified to form 6 highly chitinous plates called teeth. This 6-toothed grinding machine violently crushes the stored food particles into microscopic pieces, preparing them for chemical digestion in the midgut. But to survive the exams, you must memorize exactly what happens at the biological borders, the junctions between these gut sections.
The junctions are the memory anchors. At the exact junction where the foregut meets the midgut, there is a ring of 6 to 8 blind, hollow tubules called hepatic chica. Their specific job is to secrete powerful digestive juices directly into the midgut to chemically break down the crushed food.
Then, further down the tract, at the exact junction where the midgut meets the hindgut, there is another ring of structures. These are 100 to 150 incredibly thin, yellow-colored filamentous tubules called Malpighian tubules. These have absolutely nothing to do with digestion.
They are the primary organs of excretion. They filter the blood, extracting nitrogenous wastes. You have to lock this pairing in your mind. Hepatic chica equal digestion at the foregut-midgut border. Malpighian tubules equal excretion at the midgut-hindgut border.
Connecting those precise structures to their specific junctions is critical. And speaking of the Malpighian tubules and excretion, we need to discuss the biochemical strategy of the cockroach. The cockroach is uricotelic. This means it absorbs nitrogenous waste from its blood and chemically converts it into uric acid, which is then excreted out through the hindgut.
Now, why uric acid? As we discussed with the amoeba, biology is about solving physical problems. If a fish produces toxic ammonia waste, it can just constantly flush it out into the surrounding ocean because water is unlimited. Mammals convert ammonia into urea, which is less toxic but still requires a fair amount of water to excrete as urine.
But a cockroach lives in dry environments. Water is its most precious resource. Uric acid is almost entirely insoluble. It can be excreted as a dry, solid pellet, requiring almost zero water loss.
Synthesizing uric acid costs the cockroach a massive amount of cellular energy, ATP, but it trades that food energy to perfectly conserve its internal water. It is a brilliant survival mechanism.
That evolutionary trade-off is fascinating, paying a massive energy tax just to save a few drops of water. Now, earlier, we referred to this bug as having an open flow engine. This relates to their circulatory system, which is fundamentally different from ours or the earthworms.
We have a closed system. Our blood is pressurized within a network of closed pipes. If a pipe bursts, we bleed internally. But in the cockroach, the circulatory system is open. The blood vessels are poorly developed and simply end, opening up into massive, wide internal spaces collectively called the hemocoel.
All the visceral organs inside the cockroach are not supplied by microscopic capillaries. They are just sitting directly in the hemocoel, constantly bathed in the blood, which is called hemolymph.
And because it's an open system, there is no high blood pressure. The hemolymph is slowly circulated by an elongated, muscular, 13-chambered heart situated along the mid-dorsal line of the thorax and abdomen. Blood enters the heart through small slits called ostia, is pumped forward to the head sinuses, and then flows back into the hemocoel.
But here is the critical physiological difference. The cockroach's blood does not transport oxygen. It lacks entirely any respiratory pigment like hemoglobin. Therefore, the hemolymph is completely colorless. It consists only of plasma and immune cells called hemocytes.
But wait, if the blood doesn't carry oxygen, how does the biological engine run? Oxygen is required to make ATP. How does the oxygen reach the deep internal tissues of this armored tank?
The answer is a completely separate plumbing network called the tracheal system. The cockroach has a highly branching network of hollow tubes called trachea that run throughout its entire body. These tubes open to the outside air through 10 pairs of microscopic holes called spiracles located along the lateral sides of the body.
The large tracheal tubes branch down into incredibly fine trocheoles, which physically penetrate deep into the tissues, resting directly against the individual cells. It is like having millions of tiny internal scuba tubes delivering atmospheric oxygen directly to the mitochondria, bypassing the circulatory system completely.
It is an incredibly efficient local delivery system for a small insect. However, this tracheal respiratory mechanism is exactly why we do not see cockroaches the size of dogs today. The tracheal tubes rely primarily on passive diffusion of gases.
As an insect's body volume increases, the tracheal tubes would have to become impossibly long and thick to deliver enough oxygen to the deep core tissues. In our modern atmosphere, which is roughly 21% oxygen, the physics of diffusion strictly limit how large an insect can grow before its internal cells suffocate.
Hundreds of millions of years ago during the Carboniferous period, atmospheric oxygen levels were much higher, which allowed for massive eagle-sized insects. But today, the physics of their respiratory system keep them small.
That structural limitation is incredible context. Now, we have to look at the nervous system because this anatomical layout leads to one of the most famous and frankly bizarre facts about cockroaches. Their nervous system consists of a series of fused, segmentally arranged ganglia, which are clusters of nerve cell bodies.
These ganglia are joined by paired longitudinal connectives that run along the ventral or belly side of the body. 3 ganglia are located in the thorax, controlling the legs and wings, and 6 ganglia are located in the abdomen, controlling the digestive and reproductive organs.
And this brings us to the ultimate mind-bending realization of insect anatomy. If you physically cut off a cockroach's head, the headless body will continue to live for up to a week.
I remember reading that and just staring at the page. Yeah. A whole week. When you think about mammalian biology, that is utterly impossible. If you lose the head, you instantly lose the brain. The heart stops and breathing ceases immediately. How does the insect survive?
It feels like science fiction, but it makes perfect anatomical sense once you map out their systems. In mammals, we have a highly centralized nervous system. The massive brain in our head controls almost everything. But in the cockroach, the nervous system is highly decentralized.
The brain in the head, scientifically called the supraesophageal ganglion, is actually remarkably small. Its primary function is mostly just supplying sensory nerves to the antenna and the compound eyes.
The vast majority of the cockroach's neural processing power is distributed amongst those thoracic and abdominal ganglia spread out along the ventral nerve cord in its belly. Furthermore, it doesn't breathe through its mouth, it breathes through the spiracles along its sides.
So if you remove the head, you don't stop the breathing, and you don't destroy the neural networks controlling the internal organs. The body continues to respirate and function on autopilot.
The cockroach will eventually die, not from massive neurological trauma or suffocation, but simply from dehydration or starvation because it no longer has a mouth to drink water.
It's an unnerving level of decentralized resilience. Before we leave the cockroach, we must mention what that small brain is actually processing: its vision. Those large compound eyes situated on the dorsal surface of the head are incredible optical instruments.
Each compound eye consists of roughly 2,000 tiny, individual hexagonal photoreceptor units called ommatidia. Because it is receiving input from thousands of separate lenses pointing in slightly different directions, the cockroach receives several different fragmented images of an object simultaneously. This is called mosaic vision.
And this mosaic vision represents another fascinating evolutionary trade-off. Because the image is fragmented across thousands of lenses, it provides very low resolution. A cockroach cannot see fine details. It couldn't read a book.
However, this multiple lens system offers incredibly high sensitivity to changes in light, particularly in the dark. If a shadow moves across even a few of those 2,000 ommatidia, the cockroach detects the motion instantly. It sacrifices high-definition clarity for an ultra-sensitive motion detection system.
This is exactly why they are such successful nocturnal scavengers, and why it is nearly impossible to step on one when you quickly turn on the kitchen light.
The ultimate nocturnal survival machine. This brings us to our final organism, taking a massive leap up the evolutionary ladder to section 4, the frog. We are specifically looking at the Indian bullfrog, Rana tigrina.
If the earthworm is the soil tube and the cockroach is the terrestrial tank, our mental map for the frog is the amphibian bridge. This is the dual-life vertebrate that physically and biologically bridges the gap between aquatic existence and terrestrial living.
The term amphibian literally means dual life, and their morphology reflects this constant transition. Unlike mammals, frogs are cold-blooded or poikilothermic. They lack the internal metabolic furnace to maintain a constant body temperature. Their internal temperature fluctuates directly with the temperature of the environment.
To survive this limitation, they utilize behavioral adaptations. They use protective coloration camouflage, changing their skin color slightly to blend in and hide from predators. When the environmental temperatures become too extreme to survive, they burrow deep into the mud and dramatically slow their metabolism.
This deep summer sleep to escape extreme heat is called estivation, and the deep winter sleep to escape freezing is called hibernation.
Their skin is also highly specialized. It is smooth, moist, and slippery due to the constant secretion of mucus. But here's the detail that completely surprised me. Frogs never actually drink water through their mouths. They absorb all the hydration they require directly through their highly permeable skin.
And that permeability is essential for more than just hydration. Their skin is heavily vascularized, packed with a dense network of blood capillaries right beneath the surface. This allows the skin to act as a primary respiratory organ.
When the frog is submerged in water, oxygen from the water diffuses directly through the moist skin into the blood, and carbon dioxide diffuses out. This is called cutaneous respiration, and it is entirely passive.
Looking at their overall body structure, it is streamlined for swimming and jumping, divided strictly into a head and a trunk. There is no neck to impede movement through water, and the adult has no tail. Their eyes are noticeably bulged outward, allowing them to see above the waterline while submerged, and the eyes are protected by a transparent nictitating membrane that acts like a built-in pair of swimming goggles to prevent water damage.
We also see clear external sexual dimorphism here, just like in the cockroach. Male frogs can be distinguished by two specific anatomical features that females lack. First, males have sound-producing vocal sacs located just behind the jaw, which they inflate to amplify their mating calls.
Second, males possess a specialized, rough copulatory pad on the first digit of the forelimb. During mating, which occurs in the slippery aquatic environment, this pad allows the male to maintain a firm grip on the female.
Let's dive deeper into their internal anatomy, because we are returning to a more familiar, closed, vertebrate system, but with some critical evolutionary variations. We start with the digestive system, which features a notably short intestine. We have to ask why. Why is a frog's digestive tract relatively short compared to a cow or even a human?
It relates directly to their diet. Adult frogs are strictly carnivores. They use their sticky, bilobed tongue to catch insects and small prey. Animal protein is relatively easy to break down and absorb. They do not consume complex, tough plant cellulose, which requires a massive, long, highly convoluted digestive tract to slowly ferment and extract nutrients.
A short, efficient intestine is perfectly suited for a high-protein carnivorous diet. For respiration on land, they don't just rely on their skin. They utilize their buccal cavity, the mouth lining along with their skin, and a pair of pink, sac-like lungs located in the upper part of the trunk.
But the circulatory system is where things get significantly more complex and physically fascinating. It is a closed system featuring a muscular heart, but unlike the highly efficient four-chambered heart of mammals and birds, the frog possesses a three-chambered heart. It has two upper atria and only one single lower ventricle.
Now, a student thinking critically should immediately spot the mechanical problem here. The right atrium receives deoxygenated blood returning from the body. The left atrium receives freshly oxygenated blood returning from the lungs and skin. If both atria contract and dump their distinct blood into one single shared ventricle, shouldn't the oxygenated and deoxygenated blood just violently mix into useless, partially oxygenated sludge before being pumped back out?
Right. If the blood mixes completely, the system would be horribly inefficient. The body would never get a pure delivery of oxygen. So how does the frog survive this single ventricle design?
The answer is fluid dynamics and internal ventricular anatomy. The inside of the amphibian ventricle is not just a smooth, hollow bowl. Its walls are highly structured with deep muscular ridges and spongy pockets called trabeculae.
When the atria contract, the deoxygenated blood and the oxygenated blood enter the ventricle, but they get momentarily trapped in these separate deep pockets, which drastically minimizes the physical mixing.
Furthermore, when the ventricle contracts to pump the blood out, the blood passes through a specialized exit vessel called the conus arteriosus. Inside this vessel is a twisted structure called the spiral valve. This valve acts like a mechanical traffic cop, actively directing the mostly deoxygenated blood toward the pulmonary arteries to go to the lungs, while channeling the mostly oxygenated blood toward the systemic arteries to go to the rest of the body.
It is an imperfect system compared to a mammalian heart, but it is a brilliant evolutionary bridge.
It is an incredible manipulation of fluid dynamics. And while we are discussing the blood, we absolutely must address the major Neet trap hidden in the frog's circulatory system. This is a classic high-yield comparative question.
In humans, our mature red blood cells are enucleated. As they develop, they literally eject their own nucleus to create maximum internal space for carrying hemoglobin. But frog red blood cells are nucleated. They retain their nucleus, making them larger and oval-shaped.
This is a critical distinction. Why do mammals eject the nucleus while frogs keep it? It comes down to metabolic demands. Mammals are warm-blooded. Maintaining a high, constant body temperature requires a massive, continuous supply of oxygen. Ejecting the nucleus allows a mammalian red blood cell to carry more oxygen and squeeze through tighter capillaries.
Frogs, being cold-blooded poikiloferms, have a significantly lower overall metabolic rate. Their oxygen demands are lower, so they never face the intense evolutionary pressure to evolve in nucleated red cells. If an exam question asks you to contrast mammalian and amphibian blood, the nucleated red blood cell is the key detail to look for. Do not forget it. Frog red blood cells possess a nucleus.
Got it. Moving to the nervous system, it is highly developed and vertebrate in design, organized into a central nervous system, the brain and spinal cord, a peripheral system, and an autonomic system. The brain is securely enclosed in a bony cranium, and importantly, there are exactly 10 pairs of cranial nerves arising directly from the brain.
But as we reach the end of the frog's anatomy, I want to focus on the culmination of the reproductive and excretory systems because they merge into a very unique, highly efficient anatomical feature.
Yes, let's trace that plumbing. In the male frog, the yellowish ovoid testes are found adhered directly to the upper part of the kidneys. The testes produce sperm, which is carried out by 10 to 12 tiny microscopic tubes called the vasa efferentia. These tubes don't just exit the body, they actually enter the kidney tissue itself and open into a specific longitudinal duct called Bitter's canal.
And this leads directly to the ultimate so what of frog anatomy, the cloaca. Bitter's canal inside the kidney communicates with the urinogenital duct. This duct exits the kidney and opens into the cloaca.
The cloaca is a single, small, median muscular chamber located in the very posterior end of the body. And this single chamber literally handles everything. Feces from the digestive tract. It gets dumped into the cloaca and exits the body.
Urine, filtered by the kidneys. It travels down the urinogenital duct into the cloaca and exits the body. Sperm from the male testes, or mature eggs from the female ovaries, they are channeled into the cloaca and exit the body during mating.
The cloaca is the ultimate all-purpose evolutionary exit door. It is the pinnacle of biological space-saving design. This cloacal structure is common across amphibians, reptiles, and birds, but it is fundamentally different from the heavily separated excretory and reproductive tracts found in most mammalian anatomy.
We have covered an immense amount of ground today, tracing the evolution of these internal mechanisms. We've gone from the microscopic sealing junctions of a single epithelial cell all the way up to the fluid dynamics of a vertebrate heart.
It is time to lock this knowledge in. We need to solidify the memory anchors. Let's do a rapid-fire sticky summary.
Excellent. For everyone listening, if you remember nothing else from this deep dive, here is what you absolutely must remember.
I'm ready. Hit me with the final anchors.
1. The epithelial junctions. Tight junctions are the Ziploc seal stopping leaks. Adhering junctions are the heavy cement holding neighbors physically together. Gap junctions are the secret tunnels for instantaneous cellular communication.
Number 2, the neural tissue trap. Neuroglia, the supporting cells, make up more than 50% of the neural tissue volume, not the neurons themselves.
3. The earthworm. The typhlossole drastically increases the internal surface area for absorption in the intestine, and their hemoglobin is dissolved directly in the blood plasma while their cells act defensively.
4. The cockroach. Hepatic caecas sit at the foregut-midgut junction for digestion. Malpighian tubules sit at the midgut-hindgut junction for excretion to save water. Anal styles equal males only. And they can survive headless for a week because of their decentralized ventral nerve cord.
5. The frog. Remember, they possess nucleated red blood cells. They have a short intestine because they are strictly carnivores, and they utilize the all-purpose cloaca for the exit of both waste and reproductive cells.
Those are fantastic concrete memory anchors to lock into place. As we wrap up this deep dive into the structural organization of animals, I want to leave you with a final provocative thought. Think back to the beautiful evolutionary trend we just traced.
We started with the simple physics problem of the amoeba being unable to grow. We saw how cells solve this by dividing the labor into 4 basic tissues.
Then we looked at the earthworm, a simple, repetitive, segmented tube perfectly designed to eat soil. Then evolution pushed outward to the cockroach, an armored, highly mobile terrestrial tank with complex sensory joints and a water-saving open-flow engine.
Finally, we reached the frog, an organism with a complex bony brain box, an internal skeleton, and the dual physiological capability to breathe underwater and on land.
Here is my challenge to you. As these organisms evolved to become larger and immensely more complex over hundreds of millions of years, how did that concept of division of labor have to shift? We went from simply specializing microscopic individual cells within a tissue to specializing entirely isolated, highly complex macroscopic organ systems that must interact perfectly.
Imagine the internal control systems, the hormones, and the neural feedback loops required to keep a mammalian symphony playing in perfect tune.
Keep diving into those complex biological mechanisms, keep mapping the systems in your head, and most importantly, keep asking why the architecture looks the way it does.